the fatty acid. This is Samir Ranjan, tutor, Department of Biochemistry, Faculty of Medicine, Sri Lakshminarayan Institute of Medical Science, affiliated to Bharat Institute of Higher Education and Research, SAMI. Yes. First, we are going to see biosynthesis of fatty acid. First introduction? First, we have introduction. What is the majority of the fatty acid proteins for the body is supplied by the diet. Main fatty acid which has been supplied to our body is by diet. Next is the excess amount of carbohydrate and the protein from the diet can be converted from fatty acid and are stored as a triglycerols in our adipose tissue. It takes place, this reaction used to take place in the cytoplasm of the cell. And the major fatty acid, which is synthesized by the de novo synthesis, is palmitic acid, which is 16 carbon saturated fatty acid. Next, we are entering into requirements. What are the major requirements for biosynthesis of fatty acid? First, we need acetyl-CoA. Second, we need NADPH. And third, we need ATP. This is very important. As I said, the requirement for fatty acid, biosynthesis of fatty acid is acetyl-CoA, NADPH, and ATP. Next, we have site. What are the different sites where this reactions takes place? First site, it is takes place in the liver. Next, it is takes place in the kidney. Third, it takes place in the adipose tissue. And finally, it takes place in the mammary gland, lactating mammary gland. The site where are liver, kidney, adipose tissue, and lactating membrane. These are the important sites where the biosynthesis of fatty acid takes place. Next, we have transportation of acetyl-CoA. Means the acetyl-CoA cannot able to transport through mitochondrial membrane. So we have different mechanism to transport of acetyl-CoA. Where acetyl-CoA? First of all, where do we get from acetyl-CoA? We used to get acetyl-CoA from pyruvate. We used to. This is the main important crossing area where mitochondrial membrane. This region is called mitochondria. This region, left hand side is mitochondria and right hand side is cytoplasm. Where mitochondria, this is acetyl-CoA. This acetyl-CoA which cannot able to cross the mitochondrial membrane. Where does we get acetyl-CoA? We used to get acetyl-CoA from pyruvate, which is glycolysis, carbohydrate. Carbohydrate synthesis used to go for acetyl-CoA. Where we get acetyl-CoA is through pyruvate. This acetyl-CoA cannot be transported through the mitochondrial membrane, where it cannot cross to the mitochondrial membrane and cannot able to reach the cytoplasm, where the biosynthesis of fatty acid used to take place in the cytoplasm of the cell. But the acetyl-CoA cannot able to cross because mitochondrial membrane doesn't have a permeability to cross the acetyl-CoA. Rather than what does it takes place? This acetyl-CoA used to combine with oxaloacetate in the TC cycle, where acetyl-CoA plus oxaloacetate where to form citrate. Citrate can able easy to cross the mitochondrial membrane, where through citrate synthase, it will form citrate will form through citrate synthase. Acetyl-CoA plus oxaloacetate to form citrate by the help of citrate synthase. This citrate can cross the mitochondrial membrane and it will form again here this citrate is formed, but citrate is no need for synthesis of biosynthesis of fatty acid. Whereas citrate will go for lysis with the help of citrate lyase, it will breaks down into acetyl-CoA and oxaloacetate, where we finally we got acetyl-CoA in the cytoplasm. And it mitochondrial membrane will not allow the acetyl-CoA through mitochondrial membrane, whereas this acetyl-CoA can combine with the oxaloacetate to form citrate with the help of citrate synthase, and it get transported through mitochondrial membrane. Finally, the citrate has to break down through citrate lyase and it will form acetyl-CoA and oxaloacetate. This is the main function or main transportation of the acetyl-CoA, where it can cross the mitochondrial membrane very easily. Finally, this acetyl-CoA enters into fatty acid synthase. Is this clear? Yes. Next, we are going for major enzyme complex. Major enzyme complex, we have acetyl-CoA carboxylase and fatty acid synthase complex, where these both enzymes are multi-enzyme complex. Acetyl-CoA carboxylase is a very important to formation of malonyl-CoA, where fatty acid synthase complex, we have a different 7 multi-enzyme complex. Let me see one by one. First, we will see acetyl-CoA carboxylase. To form acetyl-CoA, formation of malonyl-CoA, first of all, we need to form malonyl-CoA from acetyl-CoA. How it forms means acetyl-CoA will converted into malonyl-CoA with the help of acetyl-CoA carboxylase, where if you form carboxylase, we have to know that there will be usage of ABC, which is nothing but ATP, biotin, and carbon dioxide. where ATP, biotin, and carboxyl were used for acetyl-CoA carboxylase as coenzyme. This ATP will converted into ADP by giving one phosphate group to the acetyl-CoA and forms malonyl-CoA. This is very important. This enzyme or this reaction is a very important, or this enzyme acetyl-CoA carboxylase is a very important and regulatory enzyme for this entire pathway. This regulatory mechanism will be seen in the last slides. First, we have to know. Next, we have to know fatty acid synthase complex. It's a very big complex where it is been attached with 7 multi-enzyme and 1 protein has been attached here. This is very important for the UG and PG sections. First, we have ketoacyl synthase. Ketoacyl synthase is an enzyme, and we are going to see in very deeply about ketoacyl, about fatty acid synthase complex. Complex means it is mixed up some many multi-complexes, where ketoacyl synthase is the first enzyme which has been carried out. Second, ketoacyl synthase has very important compound called cysteine, where it is attached with a sulfide bond. So ketoacyl synthase will have a cysteine attached with the sulfide bond. Whereas next we have acetyl-CoA transacylase, where it is combined with ketoacyl synthase. And according next to acetyl transacylase, we have malonyl transacylase. These three are known as condensing enzyme. This ketoacyl synthase, acetyl transacylase, and malonyl transacylase is known as condensing enzyme, where ketoacyl synthase will have cysteine as amino acid to it, and also sulfur bond will be there. Along with that malonyl transacylase, we have dehydratase enzyme, enolyl reductase enzyme, ketoacyl reductase enzyme, and ACP is not an enzyme, it's an acyl carrier protein. As I told, 7 multi-enzyme with 1 protein, which is nothing but acyl carrier protein, which is very helpful. Where acyl carrier protein also will have a sulfide bond along with 4-phosphopanthenine, along with 4-phosphopanthenine. So sulfide bond will be contained by two important factors by ketoacyl synthase enzyme along with cysteine, whereas acyl carrier protein along with 4-phosphopanthenine. This is very important. To this, these enzymes are called reducing enzyme. This enzyme is called reducing enzyme. Along with ACP, we have the thioesterase enzyme. It is called releasing enzyme. This is called releasing enzyme. So condensing enzyme, we have ketoacyl synthase, acetyl transacylase, malonyl transacylase, dehydratase, enoyl reductase, ketoacyl reductase, ACP, which is nothing but acyl carrier protein, and thioesterase. Condensing enzymes are ketoacyl synthase, acetyl transacylase, malonyl transacylase, whereas reducing enzyme has dehydratase, enoyl reductase, ketoacyl reductase, acyl carrier protein, along with the 4-phosphopanthenine with the sulfate bond. The releasing enzyme is called thioesterase. This is very important. Not only here, it has been completed. We have another mirror image where it has been opposite side. We have another enzyme complex along with ketoacyl synthase. Here it will be in reversed form. ACP will be attached to the ketoacyl synthase, where sulfate bridge will be formed along with cysteine sulfate bridge and 4-phosphopanthenine sulfate bridge. Sulfate bond will be formed. So we have another section of enzyme complex where thioesterase will be in the last, which thioesterase is known as a releasing enzyme. Along with the ACP is acyl carrier protein, which is along with 4-phosphopanthenine along with sulfate bond. This sulfate bond will attach with ketoacyl synthase. This just will formed very easily. From ACP along with that ketoacyl reductase will be there. Next it will be enol reductase will be there in reducing enzyme. And finally we have dehydratase. This is the important. This reducing enzyme as I have completed now. Next we are going to condensing enzyme. Condensing enzyme. Next we have malonyl transacylase along with the acetyl transacylase. Then ketoacyl synthase. This ketoacyl synthase with along with cysteine, where a sulfate bond will be there, they will be attached with ACP. Is this clear? Yes, again I'm trying to clear it, where condensing enzyme will be having ketoacyl synthase, acetyl transacylase, malonyl transacylase, where reducing enzyme will have dehydratase, enol reductase, ketoacyl reductase, ACP acyl carrier protein, and releasing enzyme may have thioesterase. So ketoacyl synthase will have cysteine along with sulfate bond. As it is, ACP will have 4-phosphopanthenine along with sulfate bond. This 4-phosphopanthenine in reverse reaction, ACP get attached with cysteine. So thioesteres, ACP, ketosyl reductase, enol reductase, dehydratase, malonyl transacylase, acetyl transacylase, and keto synthase. This is the very important complex we have. Till now, I didn't say any functional unit. This functional unit can be divided as like this. This enzyme left hand side by condensing enzyme and reducing enzyme in the bottom side will combine to form functional unit. Combines to form functional unit. Next, we have the main important synthesis. Before entering to the main important synthesis, total we have 7 steps, 7 reactions we have. So 7 reactions we have to see one by one. First, we have to take acetyl-CoA. Acetyl-CoA will be reacting with ACP, which is acyl carrier protein, along with sulfate bond, where it get transported along with the acetyl transacylase to form ACP acetyl, and OASH will be removed out. OASH will be removed out. So finally, acetyl is formed. Acetyl is combined with ACP. This acetyl ACP again it combined or transferred to cysteine. Finally, it formed ketoacyl synthase cysteine acyl. So ketoacyl synthase will be forming here. Next, we have malonyl CoA. Malonyl CoA will combine with ACP to form malonyl transacylase, where ACP will convert it into malonyl and CoASH. So this ACP with along with malonyl will be forming here. This is the second reaction. So ACP with malonyl and CoA will be removing out, whereas malonyl CoA plus ACP combines to form with the help of malonyl transacylase, it will form ACP and malonyl, where the formed two products where acetyl and malonyl will combine, where ketoacyl synthase along with acetyl and combining with ACP with malonyl to form ketoacyl synthase, where carbon dioxide is released out to form beta ketoacyl ACP. Beta ketoacyl ACP will be an important compound where it will get formed through third reaction. Next, we have fourth one, beta ketoacyl ACP, where it will get reduced with the help of ketoacyl reductase to with the help of NADPH plus H, it converted into NADP, where it will got beta hydroxy acyl ACP. Beta hydroxy acyl ACP will get formed. So ketoacyl ACP along with the ketoacyl reductase with the help of NADPH, it will get formed to NADP, where finally product we used to get beta hydroxy acyl ACP. This is fourth reaction. And from the fourth reaction, if it water molecule is removed out with the help of dehydratase, and it will form enol ACP from beta hydroxy acyl ACP, and water molecule is gone out with the help of dehydratase enzyme to form enol ACP. This is called fifth reaction. And sixth reaction, where enol ACP with the help of enol reductase. So along with enol reductase, we need NADPH. NADPH is converted into NADP to form butyryl ACP. To form butyryl ACP. This is very important, to form butyryl ACP. And this is sixth reaction. So finally, third reaction, fourth reaction, fifth reaction, and sixth reaction. It will goes for 6 cycles, and finally, with the help of thioesterase, which is known as releasing enzyme, it will form palmitate. It will form palmitate. It's very important. I will repeat it again. First, when first reaction, when it is acetyl CoA combines with ACP to form acetyl trans with the help of acyl transacylase, it will form acetyl ACP, but with the CoASH will coming out. Then acetyl will combine or transfer to form cysteine as amino acid, where it will form ketoacyl synthase enzyme cysteine along with acetyl. This is the important. Fine. Next reaction we have same like that malonyl CoA will get combined with ACP to form with the help of malonyl transacylase. It will form ACP along with malonyl, where CoA and SH will be released out from the first reaction of the product and the second reaction of the product. First reaction of the product, I got ketoacyl synthase along with cysteine and acetyl combined with ACP and malonyl with the help of ketoacyl synthase Carbon dioxide will be releasing out and it will form beta ketoacyl ACP. It will form beta ketoacyl ACP. This is very important. And third reaction, or fourth reaction, we have as we form now beta ketoacyl ACP. Next, we have beta ketoacyl ACP will get reduced with the help of ketoacyl reductase. It will get reduced with the help of ketoacyl reductase. If the reduced is being here, it will be reduced means reduction will take place where addition of hydrogen will take place. So hydrogen will be brought by NADPH and it will give hydrogen and it will form NADP. NADPH will combine NADPH will give release the hydrogen to form NADP and it will form beta hydroxy acyl ATP where beta ketoacyl ACP is converted into beta hydroxy acyl ACP with the help of ketoacyl reductase where NADP has given hydrogen and it will form NADP or reduced to form NADP. NADPH is reduced to form NADP where again it will go for releasing of water, which is nothing but dehydration or dehydratase enzyme will helpful for releasing of water. So hydroxyacyl ACP, beta hydroxyacyl ACP will converted into enoyl ACP. This is called fifth reaction, where beta hydroxy ACP, beta hydroxyacyl ACP will converted into enoyl ACP with the help of dehydratase by removing water molecule. Next, we have enoyl reductase. The reduction also takes place here. Enoyl ACP will get reduced. If it is reduction, we know that who will comes into the action. NADPH will comes into the action. NADPH will give the hydrogen to the enoyl ACP and it will form NADP because of reduction takes place. Reduced takes place. Reduction takes place. After that, enoyl ACP with combination of a hydrogen atom, it will combine butyryl ACP. So enoyl ACP converting into butyryl ACP with the help of enoyl reductase, where NADPH is converted into NADP. This is very important. Finally, butyryl ACP is the sixth reaction. So third reaction we have seen acetyl plus malonyl to form ketoacyl ACP. Fourth, we have no ketoacyl ACP is converted into beta hydroxy ACP. Fifth, we know beta hydroxy acyl ACP is converted to enoyl ACP. Sixth, we know enoyl ACP is converted into butyryl ACP. This all together will goes for repeatedly 6 cycles. Third reaction, fourth reaction, fifth reaction, and sixth reaction. Formation of beta acyl ACP, formation of hydroxyacyl ACP, formation of enol ACP, formation of butyryl ACP. These four reactions takes place 6 times again and again to form with the help to combine with the help of thioesterase enzyme, which is called releasing enzyme to form palmitate, which is that 16 carbon atom, which is 16 carbon atom palmitate. This is the formation of fatty acid. Is this clear? Yes. Next, we are going for overall reaction. Overall reaction is nothing but one acetyl CoA, which has been entered along with 7 malonyl CoA, along with 14 NADPH, and it rotates 7 ATP to form one palmitate. 7 carbon dioxide has been released out, 6 H2O has been released out, and 8 CoA will be released out, whereas 14 NADP you used to get and 7 ADP you will get because of this important reaction. So again, we have to complete, we have to come again that acetyl CoA combines with the 7 malonyl CoA with some 14 NADPH and along with 7 ATP, we used to get overall reaction as one palmitate with 7 carbon dioxide, 6 water molecule, 8 CoA and 14 NADP and 7 ADP. Next, we have regulations. Next, we have regulation. Regulation is the important part for the biosynthesis of fatty acid, where we have two types of regulation. One is allosteric regulation, another one is covalent modification of the enzyme. This is very important. Covalent modification of the enzyme. So first, we know covalent modification of the enzyme. First, we know we have, as I told in earlier, acetyl-CoA carboxylase is the regulatory enzyme for this overall reaction. So acetyl-CoA carboxylase will be in the inactive form or it will be in active form. Acetyl-CoA carboxylase will be in the acetyl-CoA carboxylase will be in the inactive form or it will be in the active form. Let me see who are all making it active and who are all making it inactive. Main thing is acetyl-CoA has to get dephosphorylation to become an active. Inactive form of acetyl-CoA carboxylase has to go for dephosphorylation to form acetyl-CoA carboxylase. Who is helping this or who is stimulating this? This stimulation will take place by insulin. This stimulation will take place by insulin. Insulin will stimulate the phosphatase enzyme to convert dephosphorylation, where acetyl-CoA carboxylase, who is having a phosphate group, will convert it into acetyl-CoA carboxylase with the active form. That means removal of phosphatase has been takes place. Dephosphorylation means removal of phosphatase takes place. And this not only stimulated by insulin, it also be stimulated by citrate, where citrate is present in the citrate has been present where it will be present in the cytoplasm, which helps to transport the acetyl-CoA from mitochondria to cytoplasm. That citrate also will go helps to activate the acetyl-CoA carboxylase, where it requires carboxylation. Okay, then this phosphorylation, if this acetyl-CoA carboxylase, if it go for phosphorylation means adding of phosphate. Dephosphorylation means removal of phosphate. So the active form has to get inactive form by adding of phosphate, which is called phosphorylation. Who is acting here? Means ATP will donate one phosphate and give and becomes ADP, where this phosphate will get combined with acetyl-CoA carboxylase to form inactive. This reaction or inhibition will take place by glucagon and epinephrine with protein kinase enzyme. This is all clear. Let's we will revise once again along with from the transportation of malonyl. First, transportation of acetyl-CoA takes place from mitochondria to cytoplasm, where acetyl-CoA cannot get transported through mitochondrial membrane. It has some channel to go through, or it has to combine with some compound to form something to cross the mitochondrial membrane, which is nothing but it will combine with oxaloacetate. Acetyl-CoA will combine with oxaloacetate to form citrate, where with the help of citrate synthase, where citrate can able to cross the mitochondrial membrane. This citrate will get lyase with the help of citrate ligase and it will form acetyl-CoA and oxaloacetate. It will form acetyl-CoA and oxaloacetate. This acetyl-CoA can be used in the cytoplasm for the fatty acid synthesis. Next, we have two major complexes where it is acetyl-CoA carboxylase enzyme as well as fatty acid synthase enzyme complex. These both are multi-enzyme complex. Acetyl-CoA formation of malonyl-CoA from acetyl-CoA is called acetyl-CoA carboxylase enzyme, which is multi-enzyme complex, where acetyl-CoA is converted into malonyl-CoA with the help of acetyl-CoA carboxylase. If it is carboxylase enzyme is there, we have to go for ABC, which is ATP, biotin, and carbon dioxide. It's needed for this reaction. This ATP will get converted into ADP, where acetyl-CoA will get converted into malonyl-CoA. This is the important regulatory step for the reaction. So acetyl-CoA carboxylase is the regulatory enzyme in this pathway. It's a very important enzyme in this pathway. As I told, fatty acid synthase complex. Fatty acid synthase complex is nothing but it's a 7 multi-enzyme complex with one protein carrier protein will be there. As first enzyme is ketoacyl synthase will be there. And along with ketoacyl synthase, we need cysteine along with sulfate bond. And acetyl transacylase will be there and malonyl transacylase will be there. So ketoacyl synthase, acetyl transacylase, malonyl transacylase will combine to form condensing enzyme. Along with that, we have dehydratase, enol reductase, ketoacyl reductase, and ACP. Dehydratase, enol reductase, ketoacyl reductase, and ACP is nothing but acyl carrier protein. Along with acyl carrier protein, we have 4-phosphopanthetine and sulfate bond. Along with that, IACP, we have a releasing enzyme. These 4 enzymes or this 4 is called reducing enzyme. Dehydratase, enol reductase, ketoacyl reductase is called reducing enzyme. ACP is a protein. And finally, we have thioesterase, which is called releasing enzyme. So, ketoacyl synthase along with cysteine, acetyl transacylase, malonyl transacylase, dehydratase, enol reductase, ketoacyl reductase, ACP, and thioesterase. These are the very important. So ketoacyl synthase along with cysteine is been binded with along with sulfate bond. Whereas ACP is a carrier protein, it's a 4-phosphatidylene along with sulfate bond. So here reverse reaction will be there. So first we have reverse means at last we have releasing enzyme will be forming here. First thioesterase along with that 4-ACP will be there along with 4-phosphopanthidylene which has sulfate bond. This sulfate bridge between ketoacyl synthase and ACP will be binded up. After that ACP we have ketoacyl reductase. After that we have enol reductase. We have dehydratase. Along with that, we have mannan transacylase, acetyl transacylase, ketoacyl synthase, which along with cysteine and sulfate bond. These are the important fatty acid synthase complex we have. So till now, I didn't say any functional unit. We have a functional unit called this condensing enzyme and reducing enzyme together at a unit is called functional unit. Next, we have first reaction of the acetyl-CoA, where acetyl-CoA will get combined with ACP with acyl carrier protein, where acetyl transacylase along with acyl transacylase, it will form acetyl ACP along with acetyl, where with the CoA will be releasing out, CoA will be releasing out. So ACP acetyl transfer of acetyl to cysteine, where ketoacyl synthase will be forming here. So with the help of ketoacyl synthase is an enzyme which will carry acetyl group along with cysteine for the further reaction. So this is called first reaction. Next, we have second reaction. Second reaction is nothing but malonyl CoA will get combined with ACP, where it will form malonyl transacylase. To form ACP will be SH sulfate bond will be removed out and CoA with sulfate bond to form OASH, where ACP will be carried by malonyl. This reaction is carried out by malonyl transacylase, where the product of two, that is ketoacyl, first reaction and second reaction, ketoacyl synthase along with acetyl with ACP along with malonyl. With the help of ketoacyl synthase, we used to form beta ketoacyl ACP. We used to form beta ketoacyl ACP by releasing carbon dioxide. By releasing carbon dioxide. Fourth, we have beta ketoacyl ACP will get converted or it will get reduced with the help of ketoacyl reductase. If it is reductase, we have NADPH. NADPH get reduced to form NADP and it will form beta hydroxy acyl ACP. Beta hydroxy acyl ACP will get dehydratase or dehydration or with the help of dehydratase by removing of water molecule, we got enol ACP. We got enol ACP. This enol ACP will converted into, this is fifth reaction with the help of enol ACP with the help of reductase enzyme, where enol reductase enzyme will use, if it is reductase enzyme is used, then NADPH will be coming into the action. NADPH is converted into NADP to form butyryl ACP. This butyryl ACP, which is nothing but third reaction, fourth reaction, fifth reaction, and sixth reaction will come go for 6 cycles to form with the help of thioesterase enzyme. Thioesterase is a releasing enzyme to form palmitate, which is nothing but palmitate 16 carbon atom. So third reaction, which is product of first and second reaction, acetyl and malonyl to form ketoacyl ACP. Ketoacyl ACP will form beta hydroxy ACP. Beta hydroxy acyl ACP will form enol ACP. Enol ACP will form butyryl ACP. These all four reactions, it will goes for 6 cycles. Finally, it will get condensed with thioesterase by help of releasing enzyme. It will form palmitate, which is 16 carbon atom. So overall reaction we have seen. Overall reaction is 1 acetyl CoA plus 7 malonyl CoA, 14 NADPH plus 17 ATP. It will form 1 palmitate, 7 carbon dioxide, 6 H2O, 8 CoA, and 14 NADP and 7 ADP. It will form. So regulation part we have seen. As regulation part we have allosteric regulation and covalent modification of enzyme. So we have acetyl CoA will be acetyl CoA carboxylase will be in the inactive form. It has to convert it into active form with the help of dephosphorylation. This dephosphorylation will be takes place by with the help of insulin, where with the help of phosphate enzyme, phosphatase enzyme will convert acetyl CoA carboxylase to form acetyl CoA inactive form of acetyl-CoA carboxylase to form active form of acetyl-CoA carboxylase. Not only the insulin, where citrate also helps to form active form of acetyl-CoA carboxylase with the allosteric regulation. Where if we want to stop the reaction, acetyl-CoA carboxylase, where in the active form will get phosphorylated or phosphorylation to form acetyl-CoA carboxylase inactive. Who will give phosphate group? ATP will give phosphate group and it will form ADP with the help of protein kinase. Who is inhibitor? Glucagon and epinephrine will be the best inhibitor. Thank you so much.